Okay, so I think the topic that we have to discuss on today, I mean, the topic means like the question, the past paper questions. What I have just, I think from nucleic acid, right? That was our
Y-
target
Yeah,
to continue on today. All right, give me a moment.
yeah,
Just want to open a worksheet for you from there, and like, yes, from nucleic acid. Yes. So I'm going to start with you from the easy, then after we go for the moderate and then for the complex, right? So for instance, I'm going to start
okay.
with you the first question that's of about 3 mark question, and then after we'll go for further to get the complex as well. For instance, we have here like this one. Abdul, so here it is in front of you. Question is, yeah, that's a figure in front of you. Just shows the structure of the nucleotide. Identify the component of the nucleotides. This is what you need to do, and you have 3 marks for it. Structure of nucleotide. If you know the components of the nucleotide, like there are 3 components which by which their structure is formed. That is already here. E, B, C. Hmm. Okay, nitrogenous base. That's good. What's about A?
Yeah, I'm writing.
Uh, should I help you to recall?
No, I know, I'm writing.
Okay, are you okay? You're right. Phosphate group. That's good, right? So what's in C?
Yeah,
Mm, yes. Okay. Pentose sugar. That's fine. All right. So then after we can go for next point. That's for 2 more questions. So that's definitely I hope you know about these things. For instance, you would have here a question for 2 marks, and it's just the difference between how you can make it more easy, like to. I think I should directly share with you the. This is how we waste our time. That should be in the form of worksheet. Okay, state two difference between DNA nucleotide and RNA nucleotides. How you'll differentiate? Like we need just two differences. How you'll different these two? Yes, DNA nucleotide contain thymine. Okay, fine. Done.
I'm just
Okay.
writing.
Contain DNA nucleotide contain thymine whereas RNA. Okay, that's fine. This is one difference. What's the second one? Because we need two differences here.
Yeah, yeah. I'm completing the first different, like RNA nucleotide contain uracil which binds with
Yes.
adenine. Then I will. go for the
Yes.
next one.
All right, that's fine.
I can't say single stranded because they are asking about nucleotide, not actual molecule. Because so I can't say double or single stranded actually. I'm
No, no,
thinking
no, no,
about
exactly. This is just for the nucleotide. We can just it
yeah,
means like we can just differentiate the components as you have
yeah.
just talked about the nitrogenous bases. So then after we can go for the difference of you know sugar molecule like uh, yeah, one is deoxyribose. Yeah, that's fine. The sugar is deoxyribose hmm, in DNA. Yeah, that's fine. And RNA has a ribose sugar. That yeah, that's these are the differences that we should work on. Then after you have the very similar sort of another easy question. We should just it's a 4 mark question. You just need to complete the sentence using the appropriate terms, right? Your terms can be single words or short phrases. It depends. Yeah. Nucleotides link together to form dash. The bonds between the nucleotides are called dash bonds and are formed through dash and this. The phosphate group has a 5 carbon of the sugar is covalently bonded to the dis of the sugar of the different nucleotides. The resulting molecule has a strong sugar phosphate backbone. Yeah. Nucleotides link to form a polynucleotide.
Is it right?
Link to form a polynucleotide? Uh, no, I think yes, that's we can say like it's going to form the polynucleotide. That's fine. Mm. Which bond is there?
Or I can say a nucleic acid as well.
Nucleotides link together to form nucleic
And
acid. Yes,
nucleic
that's also fine. But polynucleotide is understood like once the nucleotides are going to combine to each other. So that's also not wrong. Polynucleotide is also fine because nucleotides link together to form a polynucleotide. So which nucleic acid, whether the DNA or RNA, that's not like at this point of time we can't say what was that. So it's better is polynucleotide. The bond between nucleotides are called
yeah, I'm writing this.
phosphodiester. Are you writing this one? Ah, phosphodiester bonds are formed through dash reaction. Which reaction? Mm, there are two types of reactions which used to have like condensation reaction, and the second is hydrolysis. Like once you are going to break all that, so there you are using water. So in condensation, is the water is removed from there? The water molecule is removed
Yeah,
during the process of phosphodiester formation. Yeah, that's fine. So in this reaction, the phosphate group at the 5 carbon of the sugar is covalently bonded to the dash on the sugar of the different mononucleotide. Yeah, what's that? What's in this case? Which carbon that we need to locate that carbon? We have um ribose sugar, so means the 5 carbon sugar. So at which carbon this sugar, this molecule is attached? That covalent bond is formed actually. And we should also know like nitrogenous base where it is always present on the ribose sugar. Yeah.
yeah, done.
Is this what bonded to them? Three. on the sugar of the different
Mm.
nucleotide, the resulting
Three
molecule
carbon.
three, carbon number three. Yeah. And
Yeah.
next, the resulting molecule has a stronger. Okay, fine. So then after we have one like another type of question. Like it's also simple. It's of 6 marks. So I think we can talk about it as well. Like here it is. Look at this. The bases of nucleotides link together using complementary base pairing. Complete the table below for the ribonucleic acid or RNA. So adenine type of base, the complementary base, number of hydrogen bonds in the base pair. So that's also quite simple, but you need to add 1, 2, 3, 4, 5, 6. Yeah, this is for 6 marks. Hmm. Adenine pair with it's type of base. Type of base. Yes. Fine, good. Adenine is for purine, cytosine is for pyrimidine. Yeah, that's fine. What's its complementary base now? In case of in RNA. What's a complementary base of adenine?
Uracil.
Yeah, so good. And in case of cytosine, we have good cavernine. So number of hydrogen bonds in case of adenine and uracil, that's what we have. Two hydrogen bonds. Good. Hmm. Done.
Yeah, done.
So it's for 6 mark question. Then after we why complementary base pairing is important? Yes, I think two more questions related to this. Then after we'll go for other points. For instance, we have here. mm, yeah, suppose this sort of situation is here. Abdul, this one. Look at this. You have 3 mark question. I mean, 2 marks question B and 1 mark for question C. Describe why complementary base pairing is important for the function of deoxyribose nucleic acid and state why the DNA molecule is described as antiparallel. First, tell me why it is important, why the complementary base pairing is important for the function of deoxyribose. Hmm? What's your name by Abdul? Each base pair, each base a specific. Hmm? Which is complement. Complementary to a specific base. Hmm? Each base a specific structure which is complementary to a specific base structure. What do you mean by this? It's quite ambiguous.
To
Question
the
is
other
why?
two.
Why it's important for the function of deoxyribose. That's the important thing that we need to know about it. Uh, why complementary base pairing? We are not talking about the word complementary base pairing. Why it's important for the function of the DNA? I think no, it's not giving you the proper answer. Um, you need to add 2 points. So is this like we should say like you should think about in the sense of its transcription and translation. But definitely, what's
Oh,
its
I got
function?
it.
What's the function of DNA? Function of the DNA is that to be transcribed and then to be translated by in the from messenger RNA and this and that. So it means like these base pairings will help in transcription translation. In what sense? This is what you need to elaborate here. You just need to add 2 points. The base pairing will help to determine. Good. All right, that's fine. The base pairing will help in to determine the sequence of bases which determines. You should write both of these points separately because it's of 2 mark question. First, talk about the replication process. Yeah, that's this is one point that you have added. It is in that sense of in case of transcription and translation. And the second thing is because it has to replicate as well. So we need to add that point as well in the form of replication. So like it ensures you would say like accurate replication of the DNA. Why how it's ensuring this all just by having but just by matching the specific base pairs like adenine with thymine, cytosine with guanine. So this is how we would say like it ensures accurate replication. And as in case of transcription translation, we always say like what it allows information to be transferred correctly, like I would say from during the process like DNA replication or transcription, because DNA has to be converted into messenger RNA for the amino acid sequence. So its correct transformation is also important. So yes, accurate copying and information
Yeah.
transfer.
So is it complete in the base pairing will have to determine the sequence of amino acids. Uh,
Accurate replication of DNA. Hmm. Yes, fine. Because in second case, it ensures the accurate replication of DNA. You can also add bit like by matching how. So there should not be like any ambiguity there. Like you should complete this sentence by matching specific base pairs. And in bracket you can write adenine with thymine, cytosine with guanine, because we are talking about the DNA. Yeah, good.
yeah.
Yeah, that's fine. Now it's correct. Now you can get the 2 marks here. All right. So then after now you can come on this like so state why DNA molecule is described as anti-parallel. Yeah. It's a one mark question, Bindice. You just need to add 1 point here. Like state why DNA molecule is described as. We are not talking about why or any other question. That's simple. Like, what do you mean by this anti-parallel? One strand is opposite to Okay. One strand is 180 opposite. 180
No,
opposite.
I'm right. No, no, actually, I'm writing it
Oh,
one
okay,
second.
okay, fine. You can correct this then after. All right.
Yeah,
One strand is one. Are you writing
180
one
degree,
180 degree
180
opposite
degree.
to the other strand? This is what you're saying. You can also add bit to make it more impactful just by saying one strand is running from 5 and to 3 and the other strand is running from 3 and to 5 and like uh if you can further short this from your side so you can, but the context is like you have to mention this point there, right? Like you could just said one runs 5 to 3 and other 3 to 5, a very short, not uh much detail. One runs 5 to 3 and other 3 to 5. All right. Yes, one strand runs from 5 to 3 and no, that's an additional information, Abdul. I mean, the first that you had already added before that. That should be the part of it.
Oh,
Yes, you should not remove that. This is just the additional information that you are giving at the end of that. As you said in first case,
okay.
that the two strands are and two quite I mean opposite to each other, as you said, at 180 degrees and at 180 degree. So then after you can add this point here. One strand runs from 5 to 3 and other from 3 to 5. Yes. Your answer is, I mean, it's completed now. Oh, you're fine. Then after we can go for to talk about like yes, its level is getting increased. The next question is I think I should show you here. As for I think I should place a worksheet, bit a worksheet. Like here you have some questions related to this. Suppose this one. Sorry. Uh yeah, I'm not able to move it. Yes, this one. So here you have that shows the steps of the DNA replication. You can see here the DNA double helix is unbound to separate the two DNA strands. Base free nucleotides are attracted to and attached to the exposed bases. The new nucleotides are linked to form a new strand. The original strand and the new strand join together to form a new DNA molecule, as we know the semi. If that question is there, oh yes, sir, so already. So describe the enzymatic reaction which occurs in step A. Describe the enzymatic reaction which occurs in step C, and state why this process of DNA replication is described as semi-conservative. Hmm. Enzymatic reaction which occurs in step A. DNA helicase breaks the hydrogen bonds between the two strands of DNA. Okay. And describe enzymatic reaction which occurs in step C. Linked together to form. Hmm. Good. So, Abdul, do you have the access of the portal that we have on from our
Yeah, yeah,
institute? Okay,
yeah, yeah,
all right. It means like if I upload anything there, so it's easy for you to download and then to solve it, to re-upload that again. That's fine.
yeah.
Describe describe the enzymatic reaction which occurs in step C. DNA polymerase joins. Hmm.
Join. I think I'm confused
Forms
between.
that actually forms the phosphodiester bonds between
Yeah, 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 yeah.
adjacent molecule and nucleotides. This is what you need to add.
Yeah. I was confused between phosphodiester or hydrogen.
No, that's the phosphodiester linkage because nucleotides are there. Because as you can see here, uh, from where you can get this idea, the new nucleotides are linked together. New nucleotides. So new nucleotides are always linked together by a phosphodiester linkage. State why this process of DNA replication is described as semi-conservative.
Yeah.
Each DNA molecule contain one new. I think you should write this properly. Like the answer should be in a way like one original or parental strand, right? Contains one original strand. Uh, you can add even in a bracket parental strand and one newly synthesized strand. You are right. You know the concept, but I'm just giving you the words that you have to use properly while answering this question. So each new DNA molecule contains one original strand and one newly synthesized strand. All right? Yes. One newly synthesized and one original or parental strand. That's great. This is what you should answer. Then after, I think we have another very you should link question to this topic. That is, it's a 4 mark question. Describe continuous and discontinuous replication. So this is what you can add here, like this one. Here, figure 3.1 that is already given you up there. So that this doesn't show the differences between continuous and discontinuous replication. You just need to describe continuous and discontinuous replication, how we can differentiate this. So definitely half of the weightage that you have to give for the continuous and then half of the weightage that you should give to the discontinuous replication.
The continuous and discontinuous.
Yes. If you remember, like the process of replication where uh, at one end, like there was a leading strand and there was a lagging strand. So leading strand was actually where DNA polymerase can add nucleotides in the same direction as I would say from the replication fork is moving like. So like that was a smooth and without any interruption, right? So are you getting this? I mean, are this something new or should I explain this for you?
I think I'm forgetting it. So
Uh,
if you
you know,
explain.
yes, you know, there is proper direction of replication. If you
Yeah,
can read
three prime, 3 to 5.
3 to 5, all right. So if that is from 3 to 5, so we have anti-parallel sort of, that's DNA polymerase. No, remember, not 3 to 5. DNA polymerase can only add nucleotides in 5 to 3 direction. As you
Yeah,
said, like, this means what? The new strand is synthesized from its 5 end and to its 3 end. This is what actual, I mean, the template strand is read in the 3 to 5 direction. This is what I'm saying. Like, the template
yeah,
strand is from 3 to 5 direction. And but the new strand that you're adding there, that will be added from 5 to 3. This is what the difference. And as we know, the new strand grows 5 to 3 towards the fork, and that's a leading strand. That's a continuous in the same direction from that replication fork. However, the lagging strand, that's in opposite direction. So where if you can go to see the template, so it's not possible for the DNA polymerase to add nucleotides in the same direction as it did in case of other strand. That was actually from, for example, I would say, let me show you one thing here. Like, you have a strand here. These are opposite strands that we have double helixes there. So if one is for you is, as suppose I'm saying this is from 5 to 3 direction, and the other one is 5 prime to 3 prime. So at one case, yes, it's possible, the template strand which is from 3 to 5 direction, that's easy, because in that case, uh, the replication process would be continuous like this.
yeah,
However, because unwinding is going on from one side, that's not from the both sides. So the unwinding is
yeah,
from this side. So it means the unwinded other structure here is the unwound structure, that is 5 here now. The other strand is 5 now.
yeah,
So now it's not possible for DNA polymerase to add nucleotides from just from the 5. So it has to find the 3 somewhere, and then it will start its replication process from 5 to 3. Definitely, if there would be 3, I mean, I would say 3 prime in the strand, so then it would be possible for the DNA polymerase to add nucleotides 5 to 3, right?
yeah,
I mean, that's a discontinuous or opposite direction of the replication fork. You know, this is how the Okazaki fragments are formed. I mean, small fragments are formed at every point. Like, I should show you those small fragments so it would be easy for you to understand. Just a moment. If I should show you like this, here it is. Oh, sorry. It's an image. Um, like this one. Look at this. You can see here in this case. This is leading strand. That's quite easy because the direction is 3 on this side. And we know the DNA polymerase always add nucleotides from 5 to 3 directions. So there is no discontinuity or something like that. That's very good. That's going smooth. However, from this replication fork, we can see like on this side, there's a 3 and. So definitely DNA polymerase is not going to add uh, nucleotides from 3. It has to find the 5 prime somewhere, right? So like from that side, it's going to add like nucleotides in this way: 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime. Because DNA polymerase always work from 5 prime to 3 prime direction. However, in the other strand, we can see this is started from the 3 prime. So it's not possible for DNA polymerase to add nucleotides at point of 3 direction, because that's not the programming of the DNA polymerase. So it has to find somewhere the 5 prime and then start adding. Are you getting this point?
You know that in the lagging strand, I know one end is the three end, but other why the other is three as well.
Which other? Which one? No, this tree is not. This tree is this lagging strand. This is for lagging strand. This tree is for lagging strand. Definitely,
So,
it should be. Why it's showing
bro,
like that?
where is the five end of the lagging strand?
Okay, this is 3 2. Definitely, if this one is 3, so it's going to 5 here, right?
But it is showing three over here in the
In what
circle.
sense? This one, this lagging
Yeah,
strand is no. This is one thing more for you to understand. This is for the newly synthesized strand, which is definitely if you'll see the newly synthesized strand, just like this one. Here you can see
yeah,
the 5 is here, so it's also showing 5. This 5 is not for this strand, parental strand. This 5 is for the newly synthesized strand. For the parental strand, this it's understood if 5 is in this side, there must be 3 on this side. In a
yeah,
similar way, the parental strand, the 3 on this side, there must be 5 on this side. But what in this diagram, what is shown here, like the leading strand and the lagging strand, this is what he is trying to show you how the DNA, because DNA polymerase work on from 5 to 3. This should be like this. Okay. So
yeah.
problem is on this side, on the lagging strand. I have told you already, like, because the parental DNA is like this. It's from 3 to 5 here. Okay.
Yeah.
So definitely this lagging strand has to build the new nucleotide just opposite to the uh, parental strand. So if 5 is on this side, so definitely has to find somewhere uh, the next point from so that it can continue its uh, strand building. Like we have another case where I can show you, like I yes, it is more appropriate. Like, and you can see in this case, here it is. It's a bit confusing, but look at this. It's showing you the proper, the overall mechanism, what's going on there. Definitely, it's not as simple as we are saying. So this RNA polymerase is here that has to come to bind on this place, and this is how you can see. This is what 5 prime, and this at 5 prime, the primer RNA primer will attach. When it will attach, then after it would be the message for the for this DNA polymerase to add new nucleotides just after this primer, and it is going to do that like this. In a similar way, and again for the next fragment, there must be the RNA primer that should attach with that specific 5 prime, and then after it will be the message for DNA polymerase to add further nucleotides in, I would say, in 3 direction of the newly synthesized DNA. So this is how it's going on like this.
But why
And
the Okazaki fragments are not joining together?
this
Mm,
would join at the end. This would join at the end, like just after removing these primers are there. That's a reason. The reason is it will not continue to connect with each other just like the leading. The reason is simple. Like from here, this 5 to it comes like this point. Okay. At this point, again, because this going, it's this is unwound. The helicase is, I mean, this strand is opening in this side. So it means like it has to get back like this. This one first, then after this one, then after this one. So it will go back side, right? The way it is going to open, again, this DNA polymerase has to find some place behind the 5 direction. Then after again 5 direction, it's going in the back side. On this side, you can see like it's going like this. It is a continuous process. Are you getting this point or
yeah.
not? In this
Yeah.
case, Abdul, in this case, what you can see here, these fragments which are formed there, you can see this is the first strand that is formed here. Then after it's going for the second point, then again third, the fourth, like this is how it's going. It, it means like the new fragments are adding in this direction, it's quite different from the opposite direction, from the opposite strand, right? That's why it's leading one. It's lagging. It means like you are getting back somewhere. Lagging means you are adding new nucleotides in the back direction. Are you getting, Abdul? Yes. Anything? So if
Hello.
you yes, Abdul, are you getting this point or not? Any
Yeah, yeah.
confusion?
Yeah, now I got that the DNA polymerase goes from 5 to 3 end, but over here, on the lagging strand,
Hmm.
it goes backwards. So first it will go 5 to 3, then it will go again back from 5 to 3.
Yes, because direction is already defined by the DNA polymerase, it has to work from 5 to 3, right? So it means like it has to find the specific point there on the strand, and then after it will add the new nucleotides there. And definitely that's a complete process. Right now, if you'll go in that case, so we have to discuss the complete process of replication. But I have just told you here, based on this diagram, you can see the first we have to add the primers there. In the case of leading strand, you can see there's only one primer on the direction
Yeah,
far, on this side. You can see here it's from
yeah,
5 to 3, so first it has to be added. But in case of Okazaki fragments are the lagging strand, you can see at every new point from where you have to make a new fragment, there would be a new primer there. First the primer will add, and then after it will allow DNA polymerase to add the nucleotides there. Are you getting this point or not? So
yes,
that's a complete process, actually, right? So this is how there are small gaps between the fragments. And this is what we know, like just after completing all of these fragments, Okazaki fragments, because Okazaki was a scientist who actually discovered this all. And then there is a ligase enzyme, which is going
yeah,
to attach these fragments together, right?
yes,
Just by removing these primers from there. So primers will be disintegrated again, and the strand will be at the end of that, you will see the daughter strand will be continued like that. There's no difference between the leading strand and lagging strand, but this is just during the process of replication. Once the fragments are ligated with each other, then after there will be no difference. The new strands on both sides are quite similar. Are you getting?
yeah.
So just based on this, you can answer this question now. Describe continuous and discontinuous replication. How you'll describe this?
So DNA polymerase adds
What you
nucleotides from
listen,
5
listen.
end to.
I can give you direction for this. Suppose what you need to write here. You have to keep in mind leading strand first. There are two strands: leading strand, lagging strand. What you need to do, what you need to say, in case of like if we are saying the difference between continuous and discontinuous, uh, like I can say here, the continuous replication occurs on the leading strand, where DNA polymerase can add nucleotides in the same direction as the replication fork is moving. Like replication fork is what? The point where replication actually starts.
Yeah,
The right? So it proceeds smoothly and without interruption because the DNA polymerase follows the unwinding strand. As I mean the strand is unbound, so DNA polymerase is following the same direction. There is no difference there. However, discontinuous replication occurs on the lagging strand, where DNA polymerase adds nucleotides in the opposite direction to the replication fork. That's the difference. In the
yeah,
second case, you can see here, if this is the replication fork here, suppose this one is the replication fork on this strand, you can see. So in lagging strand, you can see the DNA polymerase is adding the nucleotides in the opposite direction. This one, so this is how. That's the actual difference. On the other side, you can see in leading strand, the arrow is going towards the replication fork. However, on the lagging strand, you can see the arrows are going out of that, away from the um, this replication fork.
Yeah.
Abdul, are you getting?
Yeah. Yeah.
So this is what you need to answer. DNA polymerase adds nucleotides from 5' to 3' strand. The continuous replication occurs in the leading strand. That's fine. And there's no interruption in it. And you should also say DNA you have to discuss about the DNA polymerase, because the primary function, actual player here is DNA polymerase. So DNA polymerase can add nucleotides in the same direction as the replication fork is moving. Are you getting?
Yeah.
And now you can get to talk about the discontinuous replication occurs, like the lagging strand, where you would say DNA polymerase adds nucleotides in the opposite direction to the replication fork. Okay?
Yeah.
And what's the consequence because of this? If the nucleotides are just added on the opposite direction, so what's the result? The result is like there will be the formation of short fragments. That's what we called Okazaki fragments, right? And which
Yeah.
are later joined by
The DNA ligase.
DNA ligase. That's what you need to add here. Because keep in mind, it's of about, what's the total marks here in this?
Oh,
4. Yeah. So you need to add at least 2 points in both cases. 5 to 3 direction. Yeah, that's fine. This is 1 point here. And second point that you can add in first case, smooth and without interruption. In second case, for other 2 marks, what you need to do, you can talk about the lagging strand, DNA polymerase addition of the nucleotides there, and last point is Okazaki fragments. So 2 points for leading strand or continuous replication, and 2 points for the discontinuous replication. So this is how you'll get the 4 marks here. All right.
yeah. Okay. Yeah. Opposite to the replication fork.
Yes. DNA polymerase adds nucleotides, not joins. Joining is just after that. Joining is just after the fragments formation. DNA
Yeah.
polymerase adds nucleotides opposite to replication fork. Then after you would say like the last point would be here. You can say this results
DNA
in
ligase.
this results in the formation of short fragments called Okazaki fragments. Formation of short fragments called Okazaki fragments, which are later joined together by the ligase. That's what you can talk about. I mean DNA ligase, right? Yeah.
Is it part of educare? Because I couldn't see this in my book.
Sorry.
Is it part of educare as well? Because I couldn't
Yes,
see it
it's a
in the book.
because it's a replication process. Replication is a complete topic in your case, in your
Yeah. Yeah.
in your educators, JC educators. So definitely, if you're talking about the replication, so continuous and discontinuous replication also part of it. So you have to discuss about it as well. Okay.
Yeah. Then I will say the DNA ligase joins
Yes.
these.
Yes. DNA ligase will join the fragments together. Okay.
Yeah.
Again, I think there would be a small worksheet based on this. I should add here for you to have some other points, like because we're talking about the nucleotides. I'm adding here right now, like this. Here's a small. Can you see it here? Is it visible to you,
Yeah.
Abdul? Oh, yeah. So genes are
Yeah.
transcribed into RNA by the process of transcription. So RNA is then translated into an amino acid. I think we have, yes, you should try to complete this. We have just 6 minutes now. So genes are transcribed into RNA by the process of transcription. RNA is then translated into the amino acid sequence by the process of translation. Describe how the genetic code controls protein structure. Describe the characteristics of the RNA and production of the transcription. This is for 3 marks question. Then after you have to complete the sentence. That's what we need to do. And the remaining things, like I'll send you a worksheet. And I mean, if I have the remaining questions, like if more, from a question bank, like to you can solve them from your side, so that we can continue to go for the next topic tomorrow. Like this would be better option.
Yeah. Yeah, genetic code determines the sequence of genes which determines the sequence of amino acid.
Hmm.
So this makes the primary structure of
Yeah,
protein.
the sequence of bases in DNA determines the sequence of codons in the messenger RNA, because first the codon is formed. I mean, first the messenger RNA has to be uh, formed or transcribed from the DNA bases. So each codon, as we know, that codes for the specific amino acid, right? What
Yeah,
it determines, which determines the order of amino acids in a protein, right? Definitely that's affecting its structure. Are you getting this point, Abdul?
yeah,
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 this is how you can answer this. Describe how the genetic code controls protein structure. So definitely genetic code is like, before that we need to talk about the transcription, because it's a process. Transcription, then translation. So the sequence of bases in DNA actually determines the sequence of codons, because codons are in messenger RNA. That's what we use this term for messenger RNA, sequence of codons in messenger RNA, because that's a code, right? Are you getting this
yeah,
point?
yeah.
So
So
this
I'm writing, I have written the answer almost. Yeah, I have written.
the genetic code determines the sequence of genes, which determines the sequence of codons in messenger RNA. Each codon determines the specific. Yeah, that's fine. Wonderful. Yeah, that's a perfect answer. That's fine. So now you can go for the next point. Describe the characteristics of the RNA and product of transcription. So, what are those characteristics that we can talk about? While if we are talking about the, like, definitely, as we know, it's RNA is a single strand, however it is transcribed from the DNA, that is, and like we can also talk about the second character, like one uracil in place of thymine. This is what we can add here, and definitely it's a complementary copy of DNA template strand as a whole, right? So it can leave the nucleus to be translated. So I mean, these are the characters. Like you can add any other, but definitely, this is what the very apparent uh, characters of the RNA, which is formed as an end product of the transcription. So definitely, that single RNA or single strand, uracil in place of thymine, complementary to DNA. Are you getting? 2 minutes
Yeah,
remaining,
actually,
Abdul. So try
should,
to
should I
complete it.
just should I just write on as a bullet points here?
Yes, yes. Right now, here, but not in your actual exam, right? There you have to write the proper sentences, because
So
we're gonna conclude
I will
now.
try. Yeah, RNA single stranded. Uh, and I'm writing in the sentence form.
Okay. RNA is a single strand, and uracil is in place of thymine. Good. And one more thing that you can add here, there's complementarity, as we know. That's the important character.
It is
Uh,
it is the copy of
Complementary.
DNA.
Complementary copy. I mean, that's not the exact copy. Copy means like something is exactly replica of that or replica, but that's not the case. That's a complementary copy of the DNA template strand. And this is for 5 marks. I think that I should give this to you. Like you should try to solve it. And yes, Abdullah has joined. Abdullah. Okay. Just after you, Abdullah, I think Abdullah Noman. Oh, okay. He has joined. During transcription, yes, 5 points. I mean, you need to add. During transcription, what's about polymerase? Which polymerase? RNA or DNA? It's transcription, remember. RNA polymerase separates the two DNA strand. Is this what we should say in this case?
DNA. No, separate DNA helicase.
During transcription, during
DNA
transcription,
helicase.
during transcription,
No,
it's
DNA helicase is helicase.
helicase is for what? That's for replication
Sep-
process. It's
no, it's
it's
same for the
how it's saying. That's
so
the
is
difference, right?
RNA helicase.
During transcription, no, not RNA helicase. There's no helicase here in this case. You're actually mixing replication with the transcription, right? So here the polymerase that separates the two DNA molecule, and one DNA strand is used for transcription. That's called template strand. This is what you can add here. Okay. So
Assalamu
the
alaikum,
other,
जैसा
sir.
so sorry for interruption. Abdurrahman की class end हो गई है sir?
Yes, sir. It's going to end because just let him complete this question.
Okay. अब्दुल्ला वेट कर रहे हैं आपके रूम में. अब्दुर्रहमान को वाइंड अप करें और मैंने अब्दुर्रहमान से बात करनी है.
All right.
ठीक है, थैंक यू सर.
Thank you.
The
Just
other
come.
strand is known as the
No DNA helicase. Abdul, just try to change it. I mean, change this into RNA polymerase.
yeah done.
It's for transcription, right? The process that synthesizes RNA from the DNA template. That's not replication.
Mm,
This is what we always talk about in replication, right? Yes.
yeah.
RNA nucleotide form dash strand.
The antisense strand.
Mm,
Antisense, yeah.
mm, coding strand, like the or others. I mean, the first one is template strand, right? Yes, you can say
Yeah.
one. I mean, that is for actually the template strand. One strand is used for the transcription called template strand, right? The other strand is known as the
Yeah.
coding strand. Pre-RNA nucleotides form the messenger RNA strand, right? And joined together by the enzyme RNA polymerase. This is what you need to keep in mind. And that's all for today. I think Abdul will continue this further. Few of the questions from in tomorrow's class, right? Let's conclude this for today and ask Abdullah to start our class now because we have already taken his 3 minutes.
Yes, I'm here.
Right.
Thank you
Oh,
so much,
yes.
sir.
Yes, Abdullah is here. Okay.
Abdurrahman, please stay here. I am actually moving you in a different room. I just want to talk to you. Abdullah, please turn on your camera and ask Safiullah to join for the class with Miss